All right, well, fantastic. It's really great to see everyone again. There was a great first day of the BME Symposium yesterday with the sessions on imaging and neural engineering. And today we could continue that trend, focusing on regenerative medicine for this session, and upcoming will be the precision health issue as well. And so it's really exciting to have everyone. I don't know, I felt it was inspiring to sort of see this combination of talks from both the faculty as well as the students. Just really impressive to see the breadth of work that's being done here at Michigan. So we want to try to stay on schedule. So just a couple of notes, as I mentioned yesterday, in terms of, for those of you who hadn't seen it, our DEI Mentorship Awards went to Chris Tosis and Brendan Baker. So congratulations to both of you. I've been also wanting to sort of just highlight some things that have been in the news with our faculty. So Rima Coleman had a paper that was recognized by the Journal of Biomedical Engineering, was selected as an Editor's Choice Award very recently. Congratulations to Rima. Jan Stegman was selected as a Biomaterial Science and Engineering Fellow. And so really a great accomplishment and recognition of his role model as in the field of biomaterials science and engineering. And then finally, noting that very recently, Brendan Baker was named a Cellular Molecular Bioengineering Rising Star recipient. And I think Ariella was a recipient of this in the past couple of years as well, too. So it's great to see this trend continue for our department.
So as we all know, human pluripotent stem cells have great potential for regenerative medicine, since damaged cell types and tissues could be regrown from patient-specific stem cells. Which requires controlled differentiation to either organized heterogeneous structures like organoids or generating homogeneous cell populations of interest, for example, beta cells. Right? However, controlling stem cells precisely has proven quite difficult, and cell fate outcomes in many cases are heterogeneous, with results varying from cell line to cell line or even from experiment to experiment. And every person working with pluripotent stem cells has their own examples, but here's one illustration, which is cardiomyocyte differentiation with the most widely used protocol. So in the bottom left, you see 6 replicates of basically the same experiment, and you can see the variability. And on the right, you see the exact same protocol with a different cell line and a huge difference in efficiency, where green here marks the cardiomyocytes and gray is just all cells. And this is just illustrating the problem. So why is stem cell differentiation so messy? I would argue that it's not inherently messy, but that we don't understand it well enough to control it. And in particular, I think we don't sufficiently understand how the cells interact with each other during directed differentiation. Because stem cells in vivo during embryonic development don't create pure populations of cells. Right? They create organized structures, and they do that by first setting up a spatially organized microenvironment that then instructs cells to do different things. And I think the problem is that we don't know exactly how they create an organized environment or how they subsequently interpret that. And for the purpose of my talk, I will focus on one particular aspect of that, which is paracrine signaling. So the question is, how do cells create endogenous signaling gradients, and how do they then interpret those to assume their fate? And while I think that this applies to all stem cell differentiation, basically, I think one of the simplest contexts in which to illustrate it and try to learn general lessons is BMP-dependent differentiation for 2 days in 2 dimensions, which involves only a single cell fate decision. And so it's more tractable than some 10-day protocol like making cardiomyocytes. And it's still very important because this first cell fate decision still precedes all subsequent decisions to more specialized cell types of interest. And so here you see differentiated cells stained for 3 different cell fate markers. And again, used at 2 different positions. And again, you see the proportions of different cell types are quite variable between positions. But what we are looking at here is not some kind of stochastic response to a uniform external signal. It's not noise. It's actually deterministic developmental mechanisms that lead to unpredictable results only because we are not controlling the initial conditions and the boundary conditions with enough precision. And we know that because if we do control those, the initial cell distribution using micropatterning, and we force cells to make disc-shaped colonies that are roughly the size of an embryo, then we get extremely reproducible differentiation with these kind of patterns where the cell fate proportions are always the same. And I will keep talking about this BMP example, but again, I want to stress that the lessons are general, and control of geometry essentially always makes differentiation more reproducible. And for example, here is the cardiomyocyte case again, where the mess on the left gets turned into the much more clean looking picture on the right. So as it turns out, the predictable patterns you get when you control geometry with BMP treated cells corresponds to the patterning of the early embryo. So micropatterned colonies form a disc-shaped epithelium of pluripotent cells, just like the pluripotent cells in the embryo, which are these blue cells, the bottom layer of blue cells here, or the lower. And everything else around it is extraembryonic cells. And when we provide BMP, which is normally coming from extraembryonic supporting cells, the pluripotent cells differentiate in all the different cell types of the embryo that in this case organize themselves in these concentric rings, in fact surrounded by a ring of extraembryonic cells. And after several years of work by several groups, we now also know that in broad strokes, the way this spatial differentiation works parallels what we know from the patterning of the early mouse embryo. And the BMP that is provided leads the cells to produce another signaling molecule called Wnt, which then induces yet a third signaling molecule called nodal. And then depending on the precise exposure to each of these signals, different cell fates are induced. And exactly how that works is not yet well understood and is to some extent what I'm talking about today. But right now the conclusion is that the messy differentiation you get when you throw BMP at standard cultured pluripotent stem cells is actually human development playing out. And if we understand it better, we can control it better and improve stem cell technology. And for me, the part I really want to understand is where, when, and how are endogenous signaling gradients generated, and how do cells interpret these gradients? And to answer these questions, we measure signaling live during differentiation in pluripotent stem cells, and we do that by monitoring the activity of the signaling pathways, so downstream of the diffusible signaling molecules themselves, but directly upstream of gene expression. And for BMP and nodal, which are two of the three key pathways involved in these early cell fate decisions that I mentioned, activation of these pathways leads to nuclear translocation of a shared signaling protein called SMAD4. And what that means is that we can measure activity through both pathways by reading out nuclear levels of a fluorescently tagged SMAD4 protein using automated image analysis. And we can visualize other pathways in similar ways that I just don't have time to discuss right now. And so with this tool, and here you see an example of the cells being activated and it moving to the nucleus. So with this tool in hand, we could look at the signaling activity during this self-organized pattern formation or differentiation in a micropatterned colony. And here you see quantification of that with blue being low signaling and red high. And on the right is a chymograph that shows signaling density in space and time. And there's a lot going on here that I don't have time to tell you about. But the key point I want to make here is that you'll notice that the spatial pattern of signaling is continuously changing in time, essentially. And what that means is that the textbook model of static signaling gradient with thresholds to explain the sulfate pattern doesn't work. You cannot explain the sulfates we observe with some static thresholds. And so the question becomes, what is the actual relationship between a changing signaling environment and the final assignment of fates, right? How do I quantitatively explain or accurately predict where the different sulfates end up based on the signaling I'm measuring? And this question is theoretically also pretty interesting, because if the whole signaling history matters for the final state, then we've actually gone from a single parameter like concentration or level to a formally infinite number of parameters characterizing the possible histories of the cell. And some of those parameters or features you could give intuitive names like timing or duration or amplitude or baseline. But each of these could potentially control gene expression, and they're not mutually exclusive. One could control one gene, one could another, and we have evidence for some of that. But how do you then determine which ones matter and for what? Right? And one way to approach this is to think of it as generic high-dimensional data, maybe not so different from, say, single-cell RNA-seq data. And if we could, scatter plot the different sulfates along different features of the signaling history, and we would see those fates separate, right, then we would know that those are the features that matter, and we could use them to predict fate. So here on the right, you see a hypothetical example where timing, but not amplitude, is what controls the fate decision. And so this is the way we are approaching this problem. But before figuring out how history instructs fate, we actually have to make sure that history is predictive of fate at all, right? Which to some extent is still an assumption. And in a first test, we very encouragingly found that even though static thresholds can't explain sulfate patterns in these micropattern colonies, signaling histories can. So we took the average signaling activity at each radius of a colony, right, because it's symmetric. And then we applied a very simple clustering algorithm, k-means clustering, to these signaling traces. And the clustering doesn't know anything about spatial structure, but we found out that for k equals 2 and 3, so 2 or 3 clusters, the cluster assignment is nevertheless completely spatially coherent. And what's more, if you compare the spatial assignment of these signaling clusters to the sulfate domains that you measure with immunofluorescence for sulfate markers, they pretty much exactly coincide. And so you can predict the sulfate pattern based on signaling alone. And I just want to stress that this is really non-trivial for several reasons. So first, it's nice that you can predict it from signaling at all. I know this is correlation, not causation, and that's what we're working on. But you can explain it from signaling. And as I said before, we know there are multiple pathways involved, so then it's still surprising that you only have to measure one signaling protein to predict pretty much all of it. And then most surprising is that if you take this naive textbook model of thresholds in a static gradient, right, there are quantitative thresholds that you cannot tell from the signaling gradient itself. Those are inherent to the cell. So if you have a purely sort of quantitative decision between two fates, you wouldn't expect to be able to predict it based on the signaling alone. And so the fact that you can means that these signaling histories fall into discrete, qualitatively different groups, which is pretty surprising. Still, right, there are limitations to what we did here because we were averaging over a colony that is actually not completely symmetric and assigning discrete fates. So we can't predict for an individual cell what it's going to become, or even get the radial distributions, like in the middle bottom, as measured by immunofluorescence. For smaller cell so there's actually a fourth cell population on the edge between green and red, where the cell numbers are so small you don't pick up on them in the average. And so there's some reasons that that we wanted to push this to the single cell level and start predicting fate for individual cells. And so here you can see that, well, I won't show you the whole video in the interest of time, but we can track signaling in individual cells over as much as 3 days of differentiation now. And then we still needed to measure the cell fate in the exact same cells to match up their signaling history with their fate. And for that, we're using a bleaching protocol where we bleach away the fluorescent proteins at the end, and then we can stain the cells again, re-image and register it, and then match up the live data with the fixed data for fate. And now we can start looking at the single cell level at how history relates to fate. However, we didn't want to immediately deal with the complication of these additional unobserved pathways. And so we simplified the problem just a little by pharmacologically blocking other pathways. So we're only looking at BMP signaling. And as you can see here, then what you get is a pretty much binary decision between two fates, where one is marked here by a transcription factor called ISL1, and one is marked by a transcription factor called SOX2. And now we can look at the signaling in individual cells that become either SOX2 positive or ISL1 positive. And you can tell these traces or his signaling histories are pretty noisy. To a large extent, we think that's still measurement noise. But if you know what you're looking at or what you're looking for, you can already see a pattern here. But it becomes really clear when you start averaging over groups of cells that have the most similar final expression of these fate markers. And now, surprisingly, what you see is that the cells that end up with one fate or another, or expression of the markers in between, we made a third group here, they don't differ by the strength of their initial signaling response to BMP. What they differ in is when their response starts turning off. And so, again, it seems here that it's all about time. It's all about timing, and it's the duration of the signal, not its level, that determines the ultimate fate, which is surprising. And so now we are trying to determine exactly at the single cell level, is there a specific time that makes the decision between these two categories, and that requires some fitting or denoising of individual histories. But even without fitting an explicit model, we can already ask how well an individual cell's history predicts its fate, and by building a classifier. And even with a simple built-in Matlock classifier like support vector machine, you can do a pretty good job predicting what a cell is going to be, which is really encouraging.
Can you wrap up in a minute, please?
Yeah. Okay, so I can actually leave it here. So there's a lot of information as well that we get for free that's not signaling, but say local cell densities, and that is interesting to relate as well. I'll just stop here and say here's a picture of my lab, and a lot of what I'm showing you is being spearheaded by my biomedical engineering PhD student Seth here in the back, in the last picture we had before COVID.
All right, thank you very much. That was a really fantastic and exhilarating talk. For the sake of time and trying to stay on schedule, I want to encourage people to ask questions through the chat for Idse, as well as potentially during the panel session. But I want to move on to our last talk, if I could, where I get the great pleasure to introduce our next speaker, Professor Ariella Shikanov, who is an associate professor in the Department of both Biomedical Engineering, as well as an appointment in the Departments of Obstetrics and Gynecology. Well, first off, I'll say Ariella is an amazing instructor. She's won sort of been nominated for the Golden Apple Teaching Award, and at the same time is a fantastic mentor as well, too, having won the Outstanding Undergraduate Research Opportunity Program Mentor Award in 2014. At the same time, while she's been great as a mentor and instructor, I think we're here to hear more about her science and she has focused her laboratory around the treatment of premature ovarian insufficiency, which are unfortunately all too common for just intrinsically in some patients, but also as a consequence of some of the therapeutics that are out there. And so I will let Ariella tell you about her work working with ovarian follicles and their maturation and therapeutic opportunities. Please.
Well, thank you so much for such a kind introduction. And I think I fit just perfect between development told you just by Idse and aging told you by Carla. So I'm just right in between. We'll be talking about reproduction and why is it important. So my lab is developing functional and tunable biomimetics for reproductive tissue engineering. I would like to shout out to my lab. They're absolutely amazing people, and also thank the funding sources. So my research is all about fertility. Young women who face sterilizing treatments usually don't have a lot of options. If they're healthy and they can undergo ovarian stimulation, they can potentially choose fertility preservation in terms of cryopreserving their eggs or fertilized eggs such as embryos. Unfortunately, a lot of people cannot really benefit from this. So if you think about women who have breast cancer, they cannot undergo ovarian stimulation because it takes a lot of hormones and it can make their disease worse. Post-pubertal and prepubertal girls don't really have functioning ovaries, so there is nothing to be stimulated. So for these people, we're trying to develop options so they can preserve their fertility and also ovarian endocrine function. So when you think about ovary, many people think about just fertility. Ovary is not just fertility. Ovary is also responsible for secreting most of the sex hormones such as estradiol, testosterone, androstenedione, progesterone. And once this organ gets poisoned by chemotherapy or radiotherapy, there is really loss of both fertility and ovarian endocrine function. So aiming to preserve this, the idea is before the treatment, before the exposure to toxicants, you would remove the whole organ, and then you can section it into small cortical strips or fragments of ovarian tissues that would have inside ovarian follicles. And then these strips can be frozen, and then potentially these follicles can be isolated and cultured in vitro to obtain eggs that can be fertilized and develop into a baby, or you can cryopreserve the whole strip and then transplant them later when the patient is ready to have children. So I will start with the first approach, which is in vitro follicle culture. And when we talk about ovaries, we talk about follicle. Follicle is the functional unit of the ovary. This is what's responsible for the fertility and also endocrine function. Here you have a picture of the ovary, of the follicle, and then follicles are found in multiple developmental stages in each stage. But then the basics are basically the oocyte, which is the germ cell, and it can develop into a baby once fertilized, and it's surrounded by multiple somatic cells, and these somatic cells are called granulosa cells and also theca cells. And as the follicle grows and develops, these somatic cells secrete the hormones. But then the follicle itself is the functional unit. Eggs or oocytes cannot survive without the somatic cells, and vice versa. When we talk about folliculogenesis or follicle development in vitro, we talk about all these stages. So basically most of the follicles in the ovary, and at birth, women or young girls have about 2 million follicles, and they're not renewable. Once damaged or destroyed, they cannot rejuvenate. So primordial follicles is the majority of the ovarian reserve, and once they activate and enter the growing pool, they start growing and then they become primary follicle, and you can see the oocyte gets bigger, and then there's more granulosa cells or more supportive cells around the oocyte, and it continues to grow. Secondary stage, you start developing this extra layer outside of the follicle, and then these cells also secrete hormones. Then eventually follicles reach these antral stages, which is pretty large. Then you have this fluid-filled cavity called antrum surrounding the oocyte in the middle of the follicle, and then eventually this oocyte ovulates, and whatever is left from the ovulating follicle in the ovary stays there and secretes hormones to maintain potential pregnancy. So wanting to culture these ovarian follicles in vitro, the idea is to take them out of the ovarian tissue and then plate them, culture them for a certain number of days. So in mice, it takes about 2 weeks. In humans, it can take up to 6 months. And then eventually after this growth process is finished, you would have oocytes. But then putting these follicles just on the bottom of the dish doesn't really work. They become flat, and then the granulosa cells migrate away. So this idea was first kind of innovated, pioneered in Lonnie Shea's lab in collaboration with Teresa Woodruff at Northwestern University. So the idea was to encapsulate ovarian follicles in a hydrogel, so three-dimensional soft structure that would maintain the contact between the cells, but also allow growth and expansion. Work in my lab, but the work is done in mice. We usually isolate ovaries from premature mice, and then we isolate these follicles, encapsulate them in some sort of biomaterial and culture them for days. So the first biomaterial used for 3D follicle culture was alginate. Many of you are familiar with this biomaterial. It's inert, it forms really fast, it's not toxic, and it supports follicle development. Really controlling the stiffness of these hydrogels allows you to control the follicle expansion and growth. So here you can see how follicles increase in their diameter as a function of time. And then the stiffer the hydrogel, the less growth you get. So encapsulating secondary follicles in these type of hydrogel really can get you all the way to live birth. And it was very exciting. But then most of the work was done with larger follicles, starting about 130 micron in diameter, which is secondary follicle. And when looking at the ovary at any stage, you really don't have a lot of secondary follicles. We have to focus and go back and try to culture smaller follicles, so primordial and primary follicles. So how do you do that? What we've learned that when you try and culture small follicles individually in vitro, they really don't die. So they start pretty nicely. You can see the oocyte, which is the darker circle in the middle, surrounded by granulosa cells. But then after 8 days in culture, it looks really sad. The oocyte is extruded. The granulosa cells are dark and really goes nowhere. If you co-encapsulate and co-culture multiple follicles, such as 5, they really start surviving better. They still don't grow and reach antral stages, but at least they live. And then when you put them in groups of 10, this is when the magic happens. They really survive significantly better, and they reach larger sizes, and you can actually mature and ovulate them and potentially fertilize them. So it was very exciting for us to see, to observe. And then the question was really what is happening there? Is it synergistic or additive effect? If it's just really 1 + 1 = 2, which is additive effect, and then you just have more follicles, so they grow better. So we analyzed some of the secreted factors and some of the genes as well. And what we saw that it's definitely synergistic effect. So 1 + 1 = more than 2, because when you cluster the 5x cultured follicles and 10x cultured follicles, they cluster differently. But if you just multiply the concentration of the secreted factors in the 5x, you end up here. And also, if you look at the heat map here, really the 5x follicles look significantly different than 10x follicles. So the question, what are those genes that are responsible for this synergy? So Andrea Jones in my lab is trying to figure out, she's co-culturing follicles in groups of 5 and 10. It's really pretty powerful system because, follicles in 1x don't really grow, so you cannot learn anything, they're all dead. But in follicles in groups of 5, they grow, but not as good as follicles in groups of 10. So she's running 10,000 of follicles every time, culturing them at different time points, and then analyzing the granulosa cells, and what she's seeing, then looking at the gene expression, obviously running RNA sequencing. But then, so what she's seeing that, as expected, many, so up to 3,000 genes look exactly the same. So if you look at the expression patterns between 5 and 10, you see that they're kind of, some of them are really similar, but some of them are completely opposite, right? So green is 5x and blue is 10x, and you see in the culture of follicles cultured in groups of 5, the gene expression is high, and then it goes down, and then it's completely opposite in 10x. So now she's focusing on these different gene expression patterns to identify what is it that makes the synergy work in the 10x cultures. Another approach that we're trying to take to understand what is it happening in the follicle culture is really looking at the expansion, right? So we learned that follicles can grow in alginate, but then when you want to translate it from mouse to human, we know that in human follicles reach significantly greater sizes. So when you start in mice and in human, most follicles start at about 100 to 150 micron in diameter. And then in mice, they only reach 350. But then in humans, these follicles can reach at least 2 millimeters, ideally 20 millimeters in diameter. So when you put it in alginate, alginate cannot support this kind of expansion. So we turn towards degradable hydrogels, which is PEG hydrogels, and probably many of you are familiar with this. Most of the tissue engineering community is using MMP sensitive sequences. For the follicles, we chose to use fibrin degradable sequences, mostly because plasminogen activators are very active before ovulation and during folliculogenesis. So we created similar PEG hydrogels cross-linked with fibrin sensitive sequence. And what was interesting for us to see that if you use the native fibrin sequence, which is YKNR, which is the protease sensitive sequence, the follicles degraded immediately and then basically end up at the bottom of the dish, which is not surprising because fibrin is fast degrading material. But then by just changing one amino acid in this sequence, we could really control the degradation. You can see that the follicles reach different sizes when cultured in different degradable sequences, and you can get all the way to the maturation stage and grow very nice follicles. But then yet again, when we try to culture smaller follicles, they don't really grow so well. And the major problem with culturing follicles in PEG, especially smaller follicles, that even if they grow and they reach certain sizes, the maturation rate, which you see here at M2 stage, are very low. So I'm a student in my lab, Claire, and I will not try to steal her thunder. She's giving a talk later today, but she's looking into how she can modify the extracellular matrix around these follicles. And then what she learned that when you culture follicles in PEG, there is really no deposition of the extracellular matrix proteins around the follicle. So the blue stuff is follicle. There's multiple nuclei, obviously, and you see that the surrounding area around the follicle is all black. So what she did, she modified the PEG with ECM binding sequences that allows the extracellular matrix secreted from the follicle to be sequestered. And then she sees very nice, promising results. So you can see in control, it's really black around the follicle. But then she starts modifying the PEG hydrogel with those peptides. You start seeing nice staining for the laminin, fibronectin, perlecan, and collagen. And she will show all the quantitative data. But most exciting for us was that by using those ECM binding peptides, we see significant increase in maturation stages of these follicles, which is basically the end point. So to kind of conclude the in vitro part, we're developing biomaterials to try to get to the egg, which can restore fertility in those young women or girls before puberty who basically lose their ability to have children. But then another part of this is what can we do with the ovarian tissue? So we can grow some oocytes, but then in terms of losing the endocrine function, all the hormones secreted by the ovary, it's not helping. So trying to do some work in vivo and in aiming to restore ovarian endocrine function in terms of secretion of estrogen and progesterone and other sex hormones, we're trying to build an artificial ovary. And the reason for this, instead of just implanting back the cortical strips or pieces of ovarian tissue, the risk is always reintroducing cancer cells. So if you think about leukemia patients before the age of puberty, leukemia cells are everywhere. So if you remove the organ and then implant it back, there's always a risk. So trying to address this risk and minimize this risk, what we do, we isolate individual follicles. And then you probably know that if you just infuse the cells back, the survival is not so great. So we're developing materials that can promote regeneration and kind of restoration of the ovarian endocrine function in vivo. So the first material we used was fibrin. A lot of people use fibrin, but yet again, because it degrades so fast, it couldn't really kind of remodel the environment. So we see nice follicles developing in vivo, but really the function goes down. So we moved to polyethylene glycol PEG hydrogels. And what you can see here, encapsulating ovarian follicles, there is really small, tiny follicles on day 0, and then we implanted into ovarian bursa, which is the orthotopic site in the mouse. And then after 2 weeks, you start seeing, so this is the whole implant with some follicles, and you start seeing increase in size, and the close-up shows you larger antral follicles. After 1 month, this cellular portion of the graft is increasing. You start seeing very nice antral follicles, and it goes on. So even after 2 months, you still see some remaining PEG, very nice cellular portions of those follicles developing. If you look at the function, the hormones go up and then they continue, so they maintain the endocrine function. And also, if you look at the distribution of the follicles, we implant just primordial and some primary, but as time goes by, you actually start seeing development of larger follicles. We're still maintaining the follicular pool, and also nice revascularization promise of these grafts kind of really connecting to the host. This was all done with mice follicles, tested in mice, and now Monica, who is a PhD student in my lab, she's trying to really translate this work using human follicles encapsulated in PEG hydrogel. And this is the last part of my talk, which is kind of the most exciting, I think, and the most promising, and this really, and probably it's something that would have the most impact, is focusing on prepubertal girls. So the girls that experience cancer and have to undergo sterilizing treatment. In many cases, they're sterilizing, so they also use not only their fertility, but also endocrine function. And the population of these patients is growing every day. The survival rates of childhood cancer are increasing, which is fantastic, but also the number of these patients who might need this treatment. And just looking at this photo, you obviously can notice that these two women are related. So the identical twins, right? That they don't look identical. The woman on the right, she had leukemia when she was 3, and obviously you can see that she's shorter. She's predisposed to obesity, different metabolic problems, and she is also infertile. And one of the reasons, not only, but one of the reasons for this is not having functioning ovaries when undergoing puberty. And the only treatment right now, so these girls who lost their fertility, who lost their function when they're 12 to 14 and they don't start puberty, they go to an endocrinologist, a pediatric endocrinologist, and they're prescribed hormonal replacement therapy, which was developed to treat postmenopausal symptoms. And Idse really introduced this idea of dynamic signaling, and I couldn't have said it better. So HRT just delivers steady levels of hormones that don't really interact with the rest of the body. And if you look at what's happening normally endogenously, during the cycle, so one menstrual cycle of 28 days, you have different levels. So estrogen is low and then goes up and then decreases. FSH is fluctuating. All these levels are fluctuating. So when you do something like this, such as an HRT, you really end up with inferior outcomes. And it's not surprising because ovary or sex hormones secreted from the ovary are interacting with everything. So ovaries are controlled by the HPG axis, so it's controlled by the hypothalamus and pituitary. Ovaries talk to the liver and obviously fat and muscle and bone. So when you lose this kind of crosstalk between the multiple organs, you end up with shorter gait and also predisposure to different metabolic and cardiovascular diseases. So our idea was, what if we take a piece of donor tissue, ovarian tissue, and we encapsulate it in immunoisolating device, because you cannot just implant donor tissues without inducing immune rejection. And then this tissue would sense all the hormones stimulating in the body and respond by secreting sex hormones. So the idea is it's going to be self-regulating. So you just put cells and cells do what they need to do. And donor tissue is available really plenty. So how do you build something like this? Again, we went back to the PEG idea, but you cannot just make it completely degradable, because it's completely degradable, what we've showed before, that the tissue gets revascularized and then it's going to be rejected. So the idea was to use degradable tissue because follicles expand and they grow so much and increase in size, but then it's going to be surrounded by non-degradable shell. And this is how this capsule looks like, subcutaneously in the mouse with the tissue in the middle, surrounded by the gel. And what we've been able to show that this tissue can survive when implanted in mice in this immuno-isolating capsule for up to 2 months and even longer. And then this tissue doesn't really cause any immune rejection because we cannot measure any circulating antibodies. And also mice resume cyclicity. So without anything, mice don't cycle But then when you implant these capsules with ovarian tissue, the mice resume cyclicity and they can experience normal function. So we did everything possible in mice. We showed that obviously autologous tissue works and allogeneic works, and it also can work in sensitized mice that we previously sensitized with the non-encapsulated graft. And then the question was, okay, what do we do next? So the next one was to apply for a pilot grant and really do some work with the non-human primates. And when you move from mice to non-human primates, there's always, there's size, obviously, there is different function, the physiology is slightly different. So the first question was to actually just see if the autologous transplantation, if just monkey tissue encapsulated in this capsule can function. And then the second question was, if we actually take allogeneic graft and we encapsulate it, does it continue to grow and also doesn't get rejected? Because non-human primate or human tissue reaches much greater sizes. So if the capsule breaks and the tissue gets exposed, it would get rejected. So this is how the capsules looked before we implanted them. We were very fortunate to have 3 non-human primates, 3 monkeys. They were prepubertal monkeys, so they were young, about 3 years old. So we removed tissue, we ovariectomized them. This is how the tissue looks like. So this is purely prepubertal tissue. There's only tiny small follicles. And then you can see, so before variectomy, there is some fluctuation of hormones, but it's not too high. And then after variectomizing them, we analyzed, and some of the follicles grew and reached these antral stages. But if you look at the hormone levels, there is obviously some break because it takes for the tissue to regain functionality. And then these monkeys continued cycling, continuing showing, it's not cyclicity necessarily, but it's the same fluctuating dynamic levels of hormones as we would see if they had their normal ovaries. And then so the first experiment was with autologous tissue, and then we followed up in the same animals and implanted allogeneic grafts, and you see that these grafts continue to function and secrete hormones, which was super exciting. And then the next step was to try and do the same thing with human tissue. So we work with the procurement companies where we can purchase ovaries and reproductive tracts from young women who die in car accident and whatnot. So, and this tissue is delivered fresh on ice to our lab, and then we dissect it. We remove these, this is the whole reproductive tract, so right, cervix, uterus, fallopian tubes, ovaries. So we remove the ovaries, we remove the cortical strips from these ovaries, and then small strips, small cubes encapsulated in hydrogel and then implanted in mice. And this is how these grafts look under the skin of mice. And we try to analyze and see if the human tissue implanted in mice can actually survive and reach nice sizes. We have a pretty nice bank of human ovarian tissue if anybody is interested. So we have to analyze and really count follicles and see how it looks like. And then the whole analysis is looking into development of ovarian follicles when implanted in mice. To try to push forward and move this technology into humans, I had to create a startup company. And right now we are in the process of submitting our FDA application. So this is my last slide. I know probably over time, but yeah, if you have any questions, please.
All right, great. Thank you very much, Ariella. That was a wonderful presentation around the 3 very different approaches that really is fantastic and exciting. For the sake of time, I'm going to encourage questions to go in through the chat to Ariella. And at the same time, I can turn to Brendan and ask him if he can play the student talk, please.
Hi, everyone. My name is Christopher Davidson, a 5th-year PhD student in the Baker Lab. I'm excited to share with you all today a very quick overview of my work studying cellular communication during the self-assembly of microvascular networks. The ability of cells to communicate with each other is critical to tissue formation and function. And while there are many mechanisms by which cells communicate, one that I'm specifically interested in is how cells communicate mechanically via cell-generated forces transmitted through a fibrous ECM. This type of signaling is faster, longer range, and more directional than biochemical signaling, as the fibrous nature of native tissue supports transmission of cell forces over large distances. One biological setting where mechanical communication could play an important role is during vasculogenesis, or the de novo formation of a vascular network, as this process requires single endothelial cells to intelligently assemble to create a complex 3D interconnected structure. If fully understood and harnessed in vitro, this process presents a promising approach to engineering microvasculature, a significant challenge in the field of tissue engineering. My work has utilized a synthetic electrospun fibrous biomaterial platform that has fine control over physical matrix properties such as matrix stiffness to better understand mechanical communication and vascular network formation in fibrous settings. So first, I used our fibrous biomaterial system to develop a new model of endothelial cell network formation, finding that cell forces and low stiffness matrices play a critical role during this process. I next established a cell patterning approach to investigate mechanical communication between cells, finding that low stiffness fibrous matrices support communication and directed migration of cell pairs towards each other. And using this technique, I also identified that two important proteins, focal adhesion kinase, or FAK, and the mechanosensitive ion channel TRPV4 are critical during this process. I next extended these observations to a more translatable 3D setting and confirmed that matrix stiffness, FAK, and TRPV4 are also all required for mechanical communication and resultant vascular network formation in 3D fibrin hydrogels. And lastly, I utilized a technique established in our lab to add electrospun fibers within 3D hydrogels, finding that this additional fibrous cue enhances communication and the assembly of vessel-like networks in this setting. Thank you all for listening, and I'm happy to expand on any of these projects or answer any questions that you may have.
Hello, my name is Nick Schott, and my research focuses on the development of modular microtissues for vascularized bone tissue engineering. Critical size bone defects fail to heal spontaneously without therapeutic intervention. For this, cell-based tissue engineering strategies have been explored as a means to address the drawbacks of standard bone grafting treatments. Recent attention placed on strategies that pre-vascularize constructs to promote cellular survival after transplantation and enable an appropriate healing response. Mesenchymal stromal cells, or MSCs, are ideal for this application, given their well-documented bone-forming potential and capacity to support vessel development as a supportive perisite. Establishing MSC lineage commitments prior to transplantation is critical to enable their bone regenerative ability. However, promoting both phenotypes simultaneously is proven challenging, largely due to incompatible culturing conditions. To address this challenge, we have developed a modular biomaterial platform in which cells are embedded within discrete microenvironments tailored to drive either an osteogenic or vascular function. These are formed by combining cells within a solubilized matrix and emulsifying the mixture to generate populations of highly uniform cell-laden constructs or microtissues. This approach enables greater control of separate culturing conditions where we can independently support either an osteogenic or vascular function. We've shown that pre-culturing these microtissues in suitable medium successfully induces both osteogenesis and a perisite phenotype of MSCs. Further, their modular nature permits subsequent combination to form a dual phasic system containing osseous components with an integrated vascular network. The ability to separately control inductive cues to guide multiple cellular functions makes this microtissue approach an attractive means for generating a vascularized bone construct. And we also believe it can be viewed as a platform technology to vascularize other tissues or even generate more complex and heterogeneous constructs. With that, I'll end my talk, and I'd like to thank you for your time and attention.
My name is Nicole Friend, and I'm a fourth-year PhD student co-advised by Andrew Putnam and Jan Stegman. My thesis work focuses on the development of PEG-based microbeads for therapeutic vascularization. There's a strong need to revascularize ischemic tissues that are caused by plaque buildup and blood flow blockage as a result of cardiovascular disease. Like other researchers, we take a combinatorial approach using cells and scaffolds to create our cell-laden microbeads. These microbeads, containing endothelial and stromal cells, can be pre-vascularized in vitro to form a primitive vessel network within the beads, and injected into the site of ischemia as a minimally invasive therapeutic. Once injected, these beads can rapidly anostomose with each other as well as host vasculature to quickly form robust functional vascular networks. We have previously had success forming microbeads using fibrin, shown here on the right in red. However, a lack of control over microbead properties, including size, compaction, and aggregation during collection and culture, has motivated us to investigate the use of synthetic materials for our microbead approach. Specifically, we were interested in the use of pegnorbornine as a synthetic scaffold due to its ability to rapidly form homogeneous networks even at lower PEG densities under physiological conditions via thiolene photopolymerization. We first evaluated vascularization potential of degradable pegnorbornine hydrogels. Cells were able to form robust vascular networks that displayed markers of basement membrane deposition, shown here as collagen 4. The stromal cells also took on a parasite-like role, shown here by the expression of alpha smooth muscle actin that is closely associated with vessel structures. Our next step is to form cell-laden pegnorbornine microbeads, similar to the acellular beads shown here in green containing fluorospheres, via microfluidic water and oil emulsion, and evaluate the influence of prevascularization on the network forming abilities of these microbeads, both in vitro and in vivo.
My name is Emily Margolis, and I will discuss my thesis work on engineering hierarchical vascular networks. The motivation for this project is the need for effective vascularization strategies to treat cardiovascular diseases that result in damage to both large arteries and the smaller capillaries in nearby and downstream tissues. No current strategies, either clinical or tissue engineering, contain vasculature across length scales that can be immediately perfused via surgical anastomosis. One 3D printing strategy is nearing this goal and has yielded an anatomical perfusible vascular tree, while another biofabrication strategy has integrated mesoscale vasculature into the arterial circulation, but no studies yet incorporate vascular structures from the arterial scale down to the capillary scale in a surgically implantable construct resulting in immediately perfused capillaries and arteries. We seek to create a proof of concept for this idea using our expertise in engineering microvasculature for the capillary and mesoscale and harnessing in vivo biology for the arterial scale. Our first step was to engineer a 2-scale vascular construct of meso and capillary scale vessels generated by direct fabrication and vasculogenic self-assembly. We've determined conditions that yield close association of vessels and perivascular cells and that yield functional perfusion. The next step is to incorporate a third suturable length scale, a functional artery isolated from murine tissue, as shown in the schematic. The artery is cultured within similar cellular hydrogels, thereby yielding a 3-scale hierarchical vascular construct amenable to surgical anastomosis and immediate perfusion upon implantation. This will be tested in a mouse model of hind limb ischemia in which the femoral artery is ligated and removed, followed by implantation of the construct. We will assess the rate of construct perfusion and limb salvage compared to single-scale constructs on the arterial scale and the capillary scale, similar to clinical and tissue engineering strategies, respectively. We're very excited about this work, so if you have any questions or would like to discuss it further, please reach out to me or my advisor, Andy Putnam. Thank you.
Joint movement is essential for living a healthy and active life. However, in case of joint injury, especially in articular cartilage, it doesn't heal itself because cartilage is avascular. Continued use of joint causes progression of this injury into post-traumatic osteoarthritis, which causes debilitating pain and costs us billions of dollars in healthcare and lost wages. My thesis is focused on preventing this progression using stem cell-based cartilage repair. Upon injury, we observe an increase in inflammation in inflammatory immune cells in the knee joint. Since injury doesn't heal, we get sustained inflammation, which induces cartilage cells called chondrocytes to produce even more inflammatory cytokines, establishing a positive feedback loop and maintaining a highly inflammatory joint environment. Transcription factor RUNX2 plays an important role in mediating this pro-inflammatory response of chondrocytes. Therefore, we designed a gene circuit that allows the cells to autonomously suppress RUNX2 activity, and it improved matrix accumulation by MSC-derived chondrocytes. To mimic the injured joint environment, we cultured these MSC-derived chondrocyte pellets in conditioned media from inflammatory M1 macrophages and observed a decrease in matrix loss in RUNX2 suppressing pellets. Next, we treated these macrophages with double conditioned media from these pellets and observed that double conditioned media from RUNX2 suppressing pellets decreased the expression of inducible nitric oxide synthase, a pro-inflammatory marker, and increased the expression of CD206, an anti-inflammatory marker, indicating a phenotype shift in macrophages. Therefore, RUNX2 suppression in MSC-derived chondrocytes can be used to not only prevent matrix degradation, but to attenuate whole joint inflammation and to delay, if not prevent, onset of post-traumatic osteoarthritis. Next, we are evaluating our system in animal models of cartilage injury to validate our findings. Thank you.
Hi, today I'm going to be talking about the tumor microenvironment and cancer stem cells in ovarian cancer chemoresistance. Ovarian cancer is characterized by high rates of chemoresistance development and recurrence due to the presence of these cancer stem-like cells, which tend to be more chemoresistant and can repopulate the entire tumor after treatment. Recent research has shown that the non-cancer cells in the tumor microenvironment can enrich this cancer stem cell population and may be a critical distinguishing factor between the drug response of various ovarian cancer subtypes. However, we still lack a complete picture of how all these cell types work together to promote chemoresistance in the tumor microenvironment. In my first aim, we investigated the emergence of chemoresistance using a 3D seropassaging model that you can see here. With this model, we found increased stemness, chemoresistance, and malignant characteristics over long-term seropassage culture and following drug treatment, demonstrating the utility of this model as a tool to study the emergence of chemoresistance. In my second aim, we developed a tumoroid culture system with patient-derived cancer cells cultured with mesenchymal cells, endothelial cells, and immune cells in controlled ratios to be able to better investigate the interactions between the cancer stem cells and each of these other cell types simultaneously. Using this model, we found increased stemness, chemoresistance, tumorigenicity, proliferation, and malignant ECM signatures in the presence of all 4 cell types compared to cancer cells cultured alone. This demonstrates the potential malignant effect of the non-cancer cells in the tumor microenvironment. Finally, in my third aim, we used the same tumoroid culture system to perform a drug assay on tumoroids generated with 24 different cell compositions. We used this data to develop a machine learning model to predict drug response based on cell composition, and will attempt to validate this model using publicly available sequencing datasets in the near future. Together, these aims demonstrate the utility of our in vitro model systems in researching the development of chemoresistance in ovarian cancer. Using these models to dig deeper into the mechanisms and important factors in ovarian cancer chemoresistance, we can gain meaningful insights into the critical factors driving poor outcomes. We can then use these insights to develop new treatment options, improve clinical decision-making, and ultimately improve patient outcomes. Thanks for listening.
Fantastic. Thanks to all of our student speakers. It's really a wonderful series and inspiring set of talks. I hope that everyone here will be able to take some time out for the poster sessions later today, starting at 4 o'clock. But at the same time, I'd like to maybe transition to the panel discussion. I understand some people might need to leave, but hopefully some of you can stick around for at least a few minutes for this discussion. So if Brandon, if you're able to sort of bring people up into the panel, that would be greatly appreciated. Sort of set the stage for the panel discussion, I just wanted to sort of highlight how regenerative medicine at Michigan has been a very powerful force for many years. It's been a very large number of investigators across many departments, and it's really only recently with essentially the bioscience initiative that we've been able to have sort of a more have an opportunity for more centralized, coordinated development in terms of programs in this. And so that's really being led by Dave Cohn, who's one of the people that are leading the biosciences initiative. So I think the topic that I really want to bring to the panel, and maybe because of the biosciences, I'll let Dave take the first answer to this, but definitely want to involve all the panel members, is just really thinking about, what can Michigan do in this space for regenerative medicine? How can we really leverage the strengths that we have? And, what are the challenges that we would want to sort of rally around in order to really essentially, leverage the strengths that Michigan has in this area? And I think that really sort of rests on some of the, what are the key basic biological questions that exist, as well as some of the more technological issues that might be present as well too. So with that, I will stop talking, maybe turn it over to Dave, who can introduce us to maybe what's been happening with the biosciences program, and then turn it over to the rest of the panel members to sort of comment on some component of this.
Okay, thanks, Lonnie. The biosciences program really has 2 focal areas. Scientifically, we focused on the area of programmable biomaterials, using biomaterials as a platform to understand and catalyze regenerative processes. More generally, the other aspect of the biosciences is creating the glue that will finally create a community. Since tissue engineering, regenerative medicine, was launched as a formal field, 25 years ago. Michigan's always been in the top 5 by any metric, whether it's NIH grants, number of investigators, IP, startup companies, but it's largely been on the strength of individuals. And now we have an opportunity to glue things together. So what can, to address Lonnie's question, what can Michigan bring to the table if we dissect some of the talks today and yesterday in terms of what's needed? We need an understanding of the basic biological processes and signatures to unravel the onion of complexity for whatever physiological system is of interest. We need to come to an understanding of what we want to measure and how to measure it. Once we do that, we need to understand how do we impart control, reproducible control of the system, and ultimately make a strategic decision, because humans will never be as good a bioengineer as Mother Nature. So which of the numerous elements that we have the ability to measure and the ability to control, what are the most important ones to control to actually take something off the bench and make a product? So I think all of those elements speak to Michigan, because we can give a soup to nuts approach from very basic biology to technique and tool development by engineers to those of us that are bioengineers are hybrids. We strategically know how to use these tools, and we have the clinical potency as well as the translational machinery, such as through the Coulter Foundation, to translate these technologies both to the private sector and to the clinic. So I think Michigan brings the breadth and depth, and I think that's what is really our branding compared to our peer institutions.
Great, thank you very much, Dave. So maybe just to keep the conversation going, maybe I can try to really bring Rima Coleman into the conversation, who I think is on board. And I think one topic that really wasn't represented much in the talks today, but was with our student, is this idea of synthetic biology being potentially one of the tools by which there's an opportunity to integrate that into sort of regenerative medicine, but has not happened to a great extent. Maybe I could ask Rima to sort of comment on what she sees as either as the technological challenges and or the basic science opportunities within this.
Thanks, Lonnie. So the technological challenges, of course, is reprogramming the cells without having some tumor outputs, so you don't want the cells to overgrow. You don't want to insert your gene circuit into the wrong portion of the DNA, thereby shutting down the normal function of the cells. And of course, we have to have safe ways to deliver these. And one of the great things about the Biosciences Initiative is that we are focusing on biomaterials. And one of the areas that we can integrate both synthetic biology and biomaterials is the delivery of the gene circuits. And the last thing we really need, which more and more people are now doing, is to develop toolkits so that engineers who are not familiar with the ins and outs and the basic nitty-gritty of synthetic biology can still design a gene circuit that performs the function that it's looking for, that the engineer is looking for. And so I'm trying to work with basic scientists who are strong in synthetic biology, but really need us to tell them what types of gene circuits we need, what questions we're trying to ask, and how they can help us. And that's the big ask right now. The other is really understanding how cells respond to any of the changes that we're making, right? So, as one of our students said, it's not the magnitude of BMP that's being applied, but the actual length of time, right? And so we know that we don't have a clear understanding of how the cells are reacting in different particular microenvironments to specific treatments. And if we can't understand that, we can't target the right pathways using synthetic biology. And so I think we do have the necessary investigators and talent here to be able to integrate these. And I think that's how we should approach it.
Oh, great. I think that really plays off of some of the work presented by Idse, that these environments are very dynamic as well too. And so not only does your circuit need to sort of have the right response, but also it has to be sort of self-correcting for the timing at which it might need to be integrated or might need to be active as well.
Right, exactly.
All right, well, maybe I can now try to bring Lisa Larkin into the conversation a bit. She's been one of the people that's had a very successful time here and has really worked across from rodent models all the way up to large animals and looking at commercialization as well too. And maybe Lisa, maybe it'd be great to hear your comments about what Michigan could do or opportunities at Michigan with any of these challenges I sort of mentioned.
Right. So thank you for inviting me to be on the panel. And I actually only have like about 5 minutes because I actually have someplace to be at 2 o'clock. So yeah, I mean, one thing that Dave sort of mentioned, but we also have a really great technology transfer department here that helps individuals with ideas, translate those ideas out of their lab into, the clinical arena. And so that's been very helpful for me being at the University of Michigan and having access to that. I think with respect to the environment that I've encountered, because of the technology that I work with, I don't really have any technical issues. It's actually pretty straightforward what I do, this scaffoldless tissue engineering. For me, the big hurdle is FDA regulation and funding. So I think that, if we could bring in some more expertise in trying to translate these regenerative medicine technologies with respect to FDA regulatory, that would be a really great opportunity for the University of Michigan, and also different funding opportunities for non-academic endeavors. I think that both of those are areas that could strengthen our program. I think we do really well on the NIH front, but I think that we could develop a little bit more with respect to venture capitalist funding, et cetera, for these types of technologies.
Yeah, I know you've been in the context of Michigan, have been a pioneer with trying to connect us with Army, and I believe has also looked to investing in trying to allow the Advanced Regenerative Medicine Manufacturing Initiative to be something that we can access. But you're right, that still is a resource that's supporting a lot of different institutions, and so it's maybe a good starting point, but maybe incomplete in what we need.
I think that they're more on the technical level as well. They're excellent at matching you up with different companies that have technical expertise that you don't necessarily have in order to create a tissue foundry line around regenerative medicine technologies. But you're still going to hit hurdles. For example, we're now really in deep trying to get a cell source that we can amplify to the point where we can actually fabricate large, massive amounts of tissue. They have to be CGMP quality. They have to be FDA not approved, but they have to have a master plan that are approved for the process. So there's a lot of work I think that we still need to do with respect to moving the technologies forward. But I think Michigan has the expertise here. We have the people, and we have the labs here that can drive these technologies forward.
No, fantastic. And so maybe that's a segue, if I could use to, if Steve Feinberg is on the line, I comment him because I know he's been running a number of clinical trials and have products being progressed into the area of dentistry and dental applications. So it'd be great if we could have a chance to hear his thoughts on this as well too. And being cognizant of time, this will probably be the last question. We'll have to move on, but I want to say thanks to everybody in advance. So Steve, are you present?
I'm here, Ilani. Thanks a lot. I've been here starting tissue engineering since the mid-90s with Scott Hollister and I, with 3D printing, and it's moved forward to a spin-off company that I had with tissue regeneration systems. And then it went off into soft tissue, creating complex soft tissues for reconstructive surgery and regeneration. And I think one of the things that's frustrating us is I never felt that tissue engineering was a top priority for Michigan. We fought many times to try to move to the top, but we've always been frustrated, and I'm sure Lisa would understand that as well. We had a facility here, the Human Applications Lab. I was way ahead. It was a gene vector core, really, gene therapy mostly, and they disbanded it. And what I have found out, and I'm sure Lisa has as well, is I've had to go elsewhere to interact with people for CGMP facilities. And so we're actually developing a collaborative venture with the Mayo Clinic in Phoenix, who's going to help us fabricate our devices and move it on to commercialization. And that's something we need in Michigan. We need something in Michigan because as we move into our first in human clinical trials for the work we're doing for repairing complex soft tissues like lips, functional lips, it would be nice to do the first in human studies in Michigan. And that brings up another thing that we haven't talked about in this is marketing. Can you make money on your product? And this is, you know, one of the bastions of tissue engineering, Tony Atala, had a company called Tengion, where he made bladders, human bladders, and that went belly up. So there's a problem, and especially if you're dealing with autogenous tissue versus allogeneic. Allogeneic, you can use cell banks, things of that sort, that you have issues that you have to deal with. With autogenous therapy, it's more expensive because you're more personalized in your approach to treating individuals. My goal with your next session on personalized health. I just bring people, as I've been searching around, there's a company out of Ontario that we used to have Astrum here in Michigan, in Ann Arbor, and they used to make these cell-contained devices where everything occurred within one physical entity. And they eventually left, and then now they moved to Boston as Verigel. But there's an interesting thing that Lonza bought into, a company called Octane Biosciences that makes something called cocoons. And you can do all your CGMP work in one physical entity. And that makes possibly the work that you're trying to do now more marketable and cost effective. What would be nice to have in Michigan is to have the higher ups who are involved in billing and the business aspect to interact with us in a more intimate relation. So we know exactly where we're going with our work and how we can actually make it cost effective, that companies will take it on and institutions will bring it into as a product for treating their patients. Because I think Ariella is going to run into major problems. FDA, I've already done, I've already interacted with that as well. So that's a very complex issue that I think we have to develop more focus on.
Great, thank you so much. And so with that, I think, speaking to Dave as the biosciences leader, I think you've heard a number of great presentations today. So some great science from, embryonic and patterning to sort of, muscle stem cells to all the way up now to sort of these, manufacturing, FDA, and sort of clinical and billing challenges as well too. So Dave, you have your work cut out for you. So we look forward to working with you on that to see if we can't sort of make some headway on this and really facilitate regenerative medicine across that whole spectrum of things.
Well, we're here to have fun. Just very briefly to react to Steve and Lisa. I think it's important at the onset of a project, if one's goal is translation, it's important at the onset to get the scientific expertise and ducks in order to get a feel for the regulatory path, get a feel for the manufacturing path, get a feel for the marketing, as Steve talked about. We tend to keep our R01 hat on and triage translational projects based on science, and that's necessary but not sufficient. So if these non-scientific elements were front-loaded, and we do a lot of this in our NIH center, then there may not be shock or sticker shock when, after 5 or 10 years, you realize, the immense hurdles that exist in trying to translate. Some of those hurdles are ubiquitous. Not all of them are, unique to Michigan. So I think that it's important to glue everything together, and that's another aspect of what we want to coalesce.
All right, with that, thank you all for attending. We have another session starting in 14 minutes, so I'll let everyone sort of sign off and refresh themselves a bit, and hope to see you back very shortly. Thanks, everyone. Hope to see you back here soon.